Thank you. Thank you, FabLab for inviting me here. It's a great honor to speak here. I have to adjust my title because I'm not the only one who invented the Biohack Academy. I'm taking over from a very dear colleague of mine who's working at another institution now, Peter van Boheme, who actually set up the whole curriculum. I'm taking over from him now, but I am the initiator of the Open Wet Lab, which I also did with him. And that is also what I'm going to talk about mainly. We have a Fab Lab in Waag Society in Amsterdam. We have an Open Wet Lab, and we have a Textiles Lab. But around that, we do much more. So what I would like to present also, because this is a conference about from STEM to STEAM, the context within which we do work in the Fab Academy, in the Fab Academy, and the Biohack Academy. And the Biohack Academy in Amsterdam, by the way, is another bio academy that is currently in development in the Fab Lab network. But I think Fiore and I have some talks to do to make that a good collaboration. Also because we are using the same model of local labs with their own mentor, led by the lab in Amsterdam. And it is about fabricating your own biolab and then to learn how to run it. So that's also a curriculum that's very much in development. I'll come back to that later. But first I want to say something about what I would like to talk about in this conference, from STEM to STEAM, namely the A. And I've seen a few very interesting presentations yesterday that were also about the A of the arts, the A of arts embedded within humanities, which is asking the questions, why do you do things? That's why my presentation is called Cultures of Research, because I think what we do at Waag is also research. It's not scientific research, but it's research that you can call public research, not with public means, but with the public as its research community. And I think that is very similar to what happens in the Fab Lab network. It's pupils of all sorts, from a schooling background, but also from a private background, people that have an interest in doing these kinds of maker education activities. Let me see how this goes. Yeah. I'm not going to show the video. This is a picture of a workshop we did in the BioLab, where we teach people how to make competent cells so they understand how to do CRISPR-Cas mutations. And CRISPR-Cas is one of the newest gene editing techniques there is. And we think it's interesting also because there's a lot of controversy around the applications of this technique, to bring that to the public and to experience and research what to CRISPR with the public. That's the wrong one. So quickly, this is where we are. We are housed in that fantastic old building. It's an old guild house, so it had guilds of the bricklayers, the surgeons, and the artists. It's an old city gate in the midst of Amsterdam. We run a number of labs in there. And you already see that's not only a Fab Lab, but it's a contextualized space where there's more labs that are about code, about making, about healthcare design, and also about how to design learning. We happen to have had the situation where this painting was made by Rembrandt van Rijn, the very famous Dutch painter, because we have an anatomical theater in the building, a historical anatomical theater, where this historical collaboration between the arts and sciences happened. And also because we are not only organizing academies, we have more than just one kind of an output. We create laboratories of all sorts of issues. We assign performances. We do exhibitions, workshops, policy making, via a lot of other methods of creation. Co-creation, critical making, which is a large program we do now that is supported by the Netherlands Science Organization, and many more. That also leads to a number of different sorts of outputs. You may know this one. We fabricate a smartphone that is, by the way, made in Shenzhen, like all smartphones, but it's made under conditions that are special. Conditions that are fair, open, and inclusive. And I think these kinds of values are very important to use in the educational programs we organize, the research we organize, the events around it that we organize. As you can see, we also work a lot with arts. We have policy output and education models. I'm going to skip this. So quickly, this context. There's a lot going on, and you can look at these things. This is research, DNA printers, extrautero wombs. I think that relates to a fabbing community, because this is also maker culture. This is research understood as culture. And there are cultural reactions to this. And these cultural reactions come from maker communities that are trying to replicate laboratory equipment by reverse engineering, looking at equipment that has no patent anymore. And these are a few very fine results. From BioCurious in California, you see an open PCR. You also see an open PCR from Gaudi Labs, Bento Labs. And in Tokyo, there is someone working on a DNA printer. Now, that's very complicated and it takes him a lot of time. It's an artist with a design background. And then you also have an artistic reaction to that. And I think these artistic reactions are very interesting. It's artists that work with tissue cultures, trying to understand how you can create meat without having to kill an animal. That's the project there, the disembodied cuisine, which, by the way, inspired many scientists that are now making business of that. And many more, but I will come to those examples and go a bit more in depth in them. So here you can see what we do with the Open Wet Lab. We work with artists, designers, scientists, but very important, we work with the public as our research community. And what we do there is create kind of probes where the audience, the public, is able to, in some cases, touch very complex biotechnologies with the help of artists. And there the artist and the scientist and the designer are not the experts. It's in fact the public that is the expert in leading its daily life. I think that's very important to try to break down the expert layman barrier here, because all those technologies are meant for societal use, for societal application. So I think it's very important to include those citizen audiences. Yeah, I think that practical inclusion of citizens' audiences also creates a much better public understanding and cultural understanding of that research. Now I'm going to give you some examples of those cultures of research I'm speaking of, which forms the context wherein also our Fab Academy, our Fab Academy, and our Open Wet Lab, Biohack Academy function. Here you see quite a list of projects that were very inspirational for what we did, and all were working with artists a lot. Here again, the disembodied cuisine project of the Symbiotica lab Australia, run by Oran Kotz and Ionat Zur, with which we collaborated quite a few times. And here you can see a workshop that may seem a bit gross to you, but we've been researching here how to make meat. And what you actually can do is, if you take a few tiny cells of meat that is very fresh, you can put it on a growing medium, and you can see how there is new cell division happening. That is something we do with an audience in order to create an understanding of our meat-eating consumption, our meat-eating cultures. Here you can see how we did that with an audience. I'm going to skip a few. This is a workshop we did, Synthetic Biology and Arts, run by Howard Boland of C-Lab in London. He also worked with genetically modifying magnetic bacteria that he put in a dish, and he put a magnetic coil underneath it to try to see whether he can have an image change in working with bacteria. He was trying to make a bacterial TV by looking at how you can modify bacteria in an aesthetical way in order to create image machines with that. And he also taught that to an audience. So looking at Fior, this is a very interesting workshop for Biohack Academy. I'm also going to skip these. This is another interesting work that was done by two artists, Viet Pensenstadler and Hani Saad. I had a lot of discussions with them because taking biotechnologies into a public context also means that you need equipment to be taken into a public context. Because if you work with bacteria and you want to make probes, you want to make plates, you need to do that in a sterile form without hurting your own health and hurting the health of your experiments. So you need a sterile hood, and that's what you see here. Normally a sterile hood is something that is standing against the wall of a laboratory and has one-side entrance. So they designed one for public use that has a two-sided entrance, around which a larger audience can stand in order to see what the experiment is that is done and how to understand it. So it's also about changing the understanding of all the perception equipment that you have in a lab. Microscopes. I saw a very interesting workshop yesterday of making microscopes with paper. Making your own microscopes and making a lot of other equipment of laboratories is also part of that. What is also interesting here, and here you see Mary Magic, who also, by the way, comes from MIT. She did the arts department in MIT, but she trained herself to become a kind of a microbiologist, to research ecological questions. And what she did research was the fact that the plastics we make for packaging, for all sorts of applications, contain a chemical element called xenoestrogen. And that's what makes plastic weak and fallible. If you throw away plastic into the ecology, that weak maker is leaking out of it, affecting ecologies heavily. Making it, affecting sexual reproduction of microorganisms. So what she is doing here, and that's also very interesting, we did that a few times with an audience. Here we did it with an audience. By the way, coincidentally, a very female audience. It's self-extraction of estrogen from the human body, and doing experiments on microlife and see what the effect is on embryonic microlife, because it tends to become more feminine. That's a problem because microlife needs to reproduce, and reproduction is limited. So what Mary Magic designed was a yeast that was genetically modified. If you put it in surface water that contains too much xenoestrogens, it colors. So it is a biosensor made by an artist. I think it's also very interesting to see the reaction in Europe on the work of artists in a technological context. Together with a festival, Ars Electronica, a fantastic museum in Brussels, Beaux-Arts, we run the so-called STARTS Prize, which is awarded twice a year to artists that inspire technology development. Here you see a Japanese winner, and again, the Blacklist DNA printer made by Georg Tremmel in Tokyo. I think it's also important to see that these kinds of artists are also a very great inspiration to what we do in our academies. This is also an interesting work, again, using CRISPR, where artists are CRISPRing images into a DNA molecule. Because it's not allowed in Europe to do that in a cell, they used cell-free expression. So they modified the DNA molecule that was not within a living organism. Yet still, they could make that bull's head that is symbol to the relation between biotechnologies and agriculture. It's a bull's head from Dilmun, which is said to be the cradle of agricultural history. Now, I think it's very interesting to look at what artists can do in these maker contexts. And we're going to work very intensively, and she also helped out in our Biohack Academy. We're going to work very intensively with Spela Petric, who's trying to reinvent our relation to plants because of the ecological challenges we're facing. And I think because you're also talking about the societal challenges that relate to the work in Fab Labs, it's very interesting to see these kinds of concerns popping up in artists that have an interest to work within a Biohack Academy context and the context of biotechnologies. So Spela Petric is looking at how we can relate to plants by researching plants in a very unconventional way, creating an algorithm that thinks it's a plant and to try to train a plant to act like an algorithm. So I don't know whether this is going to work. I show you a little video. Can you play the video? Press the space bar probably. No, I'll go back. She puts a microscope in our Fab Lab underneath plant leaves, and plant leaves have so-called stomata. These are mouths that let in oxygen or let in CO2 and push out oxygen, so to do the photosynthesis. She made a stop-motion film of that and fed it to someone who is able to do lip reading. And he's trying to make language of that, as you can see. But she also fed it to an AI system that is doing lip reading. And they made you see, he has a lot of trouble there. He doesn't get further than syllables. Yet still, these are signs. Signs of plants within something you call biosemiotics. That's not per se only language, but it's signal exchange. They're filming all the mouths of plant leaves, and plant leaves have thousands underneath them to train a deep learning network in AI to try to see algorithms and patterns that we are not able to see there. there's a lot of scientists of the Hybrid Forms Lab of the Free University that research photosynthesis interested in this work. So we're going to set up a collaboration where this, again, artistic work inspires scientific work. And that's the way I want to make this point of cultures of research. I think it's very important to see maker research in what the Fab Lab network is doing, but also scientific research and artistic research in a context of complementary cultures of research. I see I'm a bit through my time. I think this was more or less the last slide. Yeah, well, no, quickly, this last point. Here you see Cecilia Respanti, who is with Fiore responsible for the Fab Academy to make a link between this very important industry of textiles that is so much present in our clothing, but also in all sorts of industrial textiles, and is very important in all sorts of industrial revolutions we've seen historically. They are trying to use Fab Lab context to innovate textile industry. Here you see them do a workshop called bacteria dyeing, using bacteria that excrete pigments for the dyeing industry, to try to see how you can change that pollutant in textile fabrication. I'm going to skip this one. Yeah. Last, my question, we can make so much with technology. We are democratizing that in a very fine way with Fab Lab networks and maker spaces around the globe. I think it's a very important question that if we can make everything, or if we can make almost anything, it's a real important question in the light of societal and ecological challenges, what to make. And that's where I want to end. And thank you for your attention.